Hi friends, my topic is molecular markers. So, what is molecular marker? It is a fragment of DNA which is associated with a certain location within the genome. And molecular markers are used in molecular biology and biotechnology to identify a particular sequence of DNA in a pool of unknown DNA. So, the first is amplified fragment length polymorphism. So, what is polymorphism? Poly means, as we know, many, and morphism is variation of gene. So, if we take a genome from two individuals, we can see variation between them or variation in sequences between them. So, this technique helps to identify sequences which are unique to the individuals. AFLP is a technique of or combination of two other techniques, that is RFLP, which is restriction fragment length polymorphism, and RAPD, random amplified polymorphic DNA. This is because we use both restriction enzyme and PCR technique in AFLP. The genomic DNA is cut or digested using restriction endonucleases, and using the PCR technique, the subset of restriction fragments are selectively amplified. This is the principle or the technique involved in AFLP. So the major difference between AFLP and RFLP is that in AFLP we use PCR for amplification, and in RFLP we use southern blotting. AFLP detects the presence or absence of restriction fragments instead of the length differences. It is powerful as it requires no previous sequence characterization of the target genome. Therefore, it is widely adopted by plant geneticists. It is also being used with bacterial and viral genomes. The main 4 steps included in AFLP is first one, digestion, and then adapter ligation, third, amplification, and fourth, electrophoresis. For digestion, we use two kinds of restriction endonucleases. One is MSC1 with a 4 base pair recognition site, and another one is ECHO R1-7 with a 6 base pair recognition site. Two different adapters are then ligated to these digested fragments. The features of these adapters are they are short and double-stranded DNA with sticky ends. Therefore, one adapter will complement to MSE1 cut end, and the other will complement to the ECHO R1 cut end. Using two new AFLP primers which have 2 or 3 selective nucleotides, the template undergoes amplification or selective amplification. In addition, the ECHO R1 selective primer is radio labeled. The PCR products produced by selective amplification is then separated on a polyacrylamide gel, which is used for separating the DNA bands. Normally 30 to 100 DNA bands can be detected by AFLP on a gel, and by repeating the second amplification with the other primer pairs, a different subset of the genome can be amplified. So this is a demonstration of the process or AFLP. Here we have taken a genomic DNA, and this is digested with a ECHO R1 and a MSC1. So here we can see that 6 base cutter, and here it is 4 base cutter. So it is we introduce the adapters which binds to ECHO R1 complement and MSC1, and they get ligated. So here we can see they are ligated. Then next we add the first primer with one selective nucleotide. Here it is A and here it is C. Only one selective nucleotide is added. In the next process, we add a second primer with three selective nucleotides. So first we added one selective nucleotide. We can see here A is added and T here and here C is added and they are base binding with G. So second primer with three selective nucleotides AAC and here AAC is added. By using selective amplification they are amplified and in gel electrophoresis they are separated. So the advantages of AFLP, it is that no need to know the sequence in the genome and there is high reproducibility and also many loci are simultaneously analyzed. By changing the selective nucleotide, different part of the genome can be analyzed. The whole genome analysis is possible theoretically. The only disadvantage is that it is a complex procedure. Applications. AFLP technology has the capability to detect various polymorphism or variations in the genome, many variations in the genome in different genomic regions simultaneously. AFLP has become widely used for the identification of genetic variation in strains or closely related species of plants, fungi, animals, and bacteria. It is also used in criminal and paternity tests and also to determine slight differences within populations and in linkage studies to generate maps for quantitative trait locus QTL analysis. Next is random amplified polymorphic DNA or RAPD. So here, as the name suggests, it is the random amplification which is done using PCR of polymorphic region of DNA. Here the amplification is not specific but instead it is random. So in this technique, the template of genomic DNA would be very large and the primers that we take would be very short, that is 8 to 12 nucleotides would be present. And these fragments will be amplified. Using PCR reaction, these fragments would be amplified and by resolving the resulting patterns, a semi-unique profile can be gleaned from a RAPD reaction. So this can be applied for identifying genetic variation and involves the use of single arbitrary primer in a PCR reaction resulting in amplification of many discrete DNA. The RAPD technology also provides a quick and efficient screen for DNA sequence based polymorphism at a very large number of loci. It is a method developed similar to PCR in 1990 and it is different from conventional PCR as it needs one primer for amplification. As we told earlier, the primer size is shorter and less specific. The primers can be designed without the experimenter having any genetic information for the organism being tested, as the genomic DNA normally has complementary sequences to RAPD primers at many locations. It has provided quick and efficient screen for DNA sequence polymorphism and normally a few loci can be amplified by one single RAPD primer. The procedure. The first step is that the DNA of a selected species is isolated. The outcome of the PCR, the quality influences the outcome of the PCR and high molecular weight, no impure template and less amount of RNA. An excess of selected deca oligonucleotide is added. So the 10 base primer for PCR and only one primer per reaction is added. So as we said, the primer is short and bind randomly on the chromosome. This mixture is kept in a PCR equipment and subjected to repeated cycles of DNA denaturation, renaturation and DNA replication. During this process, the deca oligonucleotide is paired with a homologous sequence present at different locations in the DNA. The DNA replication extends the deca oligonucleotide and copy the sequence continuous with the sequence with which the selected oligonucleotide has paired. The repeated cycles of denaturation, renaturation and DNA replication amplifies this particular sequence of DNA. Amplification takes place only on those regions of the genome that has the sequence which is complementary to the primer or deca oligonucleotide at their both ends. Several cycles of amplification amplification of DNA takes place and is subjected to gel electrophoresis. So initial denaturation for 10 minutes at 94 degrees Celsius and denaturation for 1 minute at 94 degrees Celsius. Annealing takes place at 37 to 45 degrees Celsius for 1 minute, extension for 1 minute at 72 degrees Celsius and the final extension at 72 degrees Celsius for 10 minutes. The cooling takes place at 4 degrees Celsius. The amplified DNA will form a distinct band when it is subjected to gel electrophoresis. It can be detected by staining with ethidium bromide and under visible fluorescence under UV light. So, as I have told earlier, this technique helps to see the genetic relatedness between two individuals. So here in this case, from this individual we have taken or isolated the DNA and it undergoes denaturation and random primers are attached at different locations of both the DNA. Then they are amplified using PCR. So here we can see they undergoes amplification and using PCR, and these amplified regions here also they are amplified and these regions are isolated and these are the regions which are isolated from here and they are loaded onto the gel. Similarly, this same process that we have done here is done on another individual. And here this black DNA represents the amplified region isolated from this individual. When both are run on the gel, we can see that the DNA has moved on the gel and DNA bands are formed. So regions here we can see these regions are similar to both the individuals and these regions which shows only other bands are different from each other. And thus we can identify the variation as well as the genetic relatedness between two individuals. Next is their advantages. The technique does not require a DNA probe and it involves no bloating or hybridization steps. Hence it is quick, simple and efficient. It requires only small amounts of DNA and the procedure can be automated. There is a high number of fragments and arbitrary primers can be easily purchased. Unit costs per assay are low compared to other marker technologies. The disadvantages of RAPD are that all RAPD markers are dominant. The PCR, since the PCR is an enzymatic reaction, the quality and concentration of template DNA, concentrations of PCR components, and the PCR cycling conditions influence or affect the outcome. Mismatches between the primer and the template may also result in the total absence of PCR product as well as in a merely decreased amount of the product. The lack of prior knowledge on the identity of the amplification product is also a disadvantage. And there is problems with reproducibility and co-migration. The applications of RAPD are genetic diversity or for polymorphism, germplasm characterization, genetic structure of populations, hybrid purity, genome mapping, developing genetic markers linked to a trait in question, or population and evolutionary genetics, plant and animal breeding, animal-plant microbe interactions, pesticide or herbicide resistance. Next is ISSRs or inter-symbol sequence repeats. The microsatellite sequences flank some of the regions in the genome, and such regions are called inter-symbol sequence repeats. In this method, we use a single primer which is used to amplify these ISSRs or these regions, and we get multiple amplification products which can be used as a dominant multilocus marker system for the study of genetic variation in various organisms. ISSRs are DNA fragments which contains about 100 to 3000 base pairs, which is located between adjacent oppositely oriented microsatellite regions. These ISSRs are amplified by PCR. The primers are microsatellite core sequences with a few selective nucleotides as anchors into the non-repeat adjacent regions, which contain 16 to 18 base pairs. ISSR markers are also known as random amplified microsatellites. As I told earlier, the ISSR markers are amplified using PCR using a single primer which is composed of microsatellite repeated sequence, and in some cases the primer also contains 1 to 4 base anchor at either 3' or 5' ends or at both ends, which target a subset of symbol sequence repeats and amplify the region between two closely spaced and oppositely oriented SSRs. This technique permits the detection of polymorphism in microsatellites and inter-microsatellites loci without previous knowledge of the DNA sequence. So in this representation, we can see the ISSR marker is generated from the two closely positioned SSR sequences. So the difference between this SSR sequence, SSR1 and the SSR2 sequence, is that they are the same repeat unit, but they are in different orientation. And the F primer, which is the forward primer, and the R primer, which is the reverse primer, is same or similar, but their orientation are also different. The sequence of the ISSR primer is the same as the SSR sequences, plus 1,3 nucleotides to obtain specific amplified products. The ISSR markers are the different sizes of the PCR amplified products originated from different sites of the genome. The advantages of ISSR. randomly distributed throughout the genome and shows high polymorphism. The main disadvantage is that there is a possibility of non-homology of similar sized fragment, and there is reproducibility problem also. So this is all about AFLP, RAPD and ISSR. Thank you.